All right, so in the last lecture, we learned about how nucleotides are added together to form nucleic acid polymers, and then we talked about the structure of probably the most famous nucleic acid structure in biology, the B-form double-stranded DNA helix. And just to remind you, this is a structure that is built out of two strands of DNA that are oriented anti-parallel to one another and are interact with one another through Watson-Crick-Franklin base pairs. And the resulting structure is this beautiful right-handed helix in which there's about 10.4 base pairs per turn of the helix, and the strands are, as I mentioned before, anti-parallel. And this structure places the sugar phosphate backbone on the outside and the nucleobases on the inside. And by placing all these nucleobases on the inside, and the fact that AT and CG base pairs, while orthogonal from one another, are perfectly stacked perfectly with one another, the resulting structure is highly stabilized no matter what the sequence of bases is. And so this is a rather beautiful structure that really facilitates its copying and replication. Of course, as I mentioned before, B-form DNA is just the tip of the iceberg in terms of the types of structures that nucleic acids can form. And in this lecture, we're going to talk about some of these other alternative DNA structures, DNA-RNA hybrid structures, and functional RNAs that have even tertiary structure that's much more complicated than just these helical structures alone. So let's extend what we've learned from double-stranded B-form DNA to understand these other structures that we'll encounter and why or why not they might form. So one of the first things to really appreciate is that, you know, if we take a much more nuanced look at nucleotide structure possibilities, things can be a little bit more complicated than this kind of static picture here. And so if you were to look at a nucleotide here, or nucleoside in this case, undulating around, you can see that there's actually a lot of conformational flexibility and complexity to the structure. And some of that conformational complexity could lead to different types of structures forming. And so one of the ones that we'll talk about right away is you may have noticed that the carbons in the pentose sugar were kind of undulating around. And that's because these carbons are not going to all lie in the same plane, but in fact, it's a three-dimensional structure, right? And in 3 dimensions, this sugar is what is called puckered, in that it can exist with either its C2 carbon tilted up or its C3 carbon tilted up, okay? And these two conformations, whichever one is oriented upward, is called the endo. And so we can think of the sugar of a nucleotide as existing in either a C3 endo or C2 endo conformation. So so different conformations, who cares? Well, it turns out there's a different preference between RNA and DNA for these different conformers. And so if you looked at an RNA nucleotide, remember that the 2' position of the sugar contains a hydroxyl group. And so it turns out that that hydroxyl group is going to make it so that the C2 endo conformation has steric problems with the nucleobase. And so for RNA, it ends up that the C3 endo conformer is the preferred one that you see. So that would be the C3 is up, okay? Now, in contrast, for DNA nucleotides, the 2' position is a hydrogen, okay? And so those steric problems that exist with the RNA's 2' hydroxyl don't exist for DNA. And so both the C2 and C3 endo conformations for DNA are possible, but it turns out that the C2 endo conformation is the one that predominates. Okay, so RNA, C3 endo, DNA, C2 endo. Is that pucker really going to make a big difference? Well, the answer is yes. It's going to make a substantial difference to the geometry of the structure that's formed. And to see why, you can note that the C2 endo conformation places the phosphate groups in the backbones much further apart from one another than in the C3 endo. And so when your phosphates are far apart like that, this is the conformation that you get in B-form double-stranded DNA like we learned. So there's this beautiful spacing here, and you get these big, wide major grooves like we see here, these narrow minor grooves. Now, if instead you're in the C3 endo conformation, that means that this the 3' carbon is moved up like this. And this has the effect of placing the phosphates in the sugar phosphate backbone much closer together. And the resulting structure that forms when this geometry is adopted is called the A-form DNA structure. And if you were to look at it here, you'll see that now that beautifully wide major groove has been sort of compressed down, and the structure is kind of much more compact. Okay. Now, B-form DNA, B-form double-stranded DNA is really the main structure that reigns supreme in terms of biological function. And of course, one of the reasons for this is that this is the conformation that allows proteins to access the information inside without needing to unwind the strands through this really wide major groove. So we talked about this last time. This wide major groove makes it easy for things to get in here. They can even read some of the sequence there. And it's very, because most DNA binding proteins bind through this major groove, we know that B-form DNA is the predominant double-strand DNA structure that has function. However, while A-form DNA is not really the primary biological form, there's many cases in which DNA and RNA are going to interact together in an RNA-DNA hybrid structure. And because RNA prefers the C3 endoconformer, RNA-DNA hybrids, as well as double-stranded RNA, which you'll also sometimes find in the cell, will adopt A-form structures. And so it's actually really important that DNA can form the A-form type of structure, because when it wants to interact with RNA, the RNA-DNA hybrids are going to have this A-form character. And something interesting about that as well is that it makes it easy for the cell to potentially distinguish between B-form DNA and A-form RNA-DNA hybrids, because the A-form, B-form structural differences are very significant. And so it's actually quite convenient that these structures have different geometries. Now, in addition to the sugar pucker, there is additional nuance in the nucleotide structure that we need to think about. So one of these is the conformation of the nucleobase itself. And so if you thought about that N-glycosidic bond between the sugar and the nucleobase, you know, there's no reason on paper that this version that we often like to draw, kind of with the base pointing outwards, we couldn't rotate around that bond and move the base inwards. And so when the base is kind of pointed over the top of the pentose ring, like you see here, we call that the syn conformation of the base. And when the nucleotide is pointed away from the sugar, we call that the anti-conformation of the nucleotide. And in general, I think it's pretty easy to imagine that, you know, from a steric point of view, placing your nucleobase right over the top of your sugar is not going to be that great compared to putting it away. And so in general, the anti-conformation of a nucleotide is more energetically favorable. But that isn't to say that the syn conformation doesn't exist or doesn't play a role. Now, if you're thinking about syn conformations, it is worth noting that only purines are capable of adopting the syn conformation, and that's because pyrimidines, which always have this carbonyl group like here, if you rotate this around and try and put in the syn conformation, it's just too sterically hindered to be a viable conformation. Whereas in your purines, the syn conformation, you just have this nitrogen there, and so it's not really impossible to adopt this structure. Now, where in biology are you going to see syn nucleotides? You know, to be honest, not very often. However, syn conformation of purines is observed in a very rare and unusual alternative DNA structure called zDNA. zDNA is sort of the black sheep of nucleic acid structures. Unlike A-form and B-form, which are right-handed, zDNA is a left-handed structure, and every other base in a stretch of zDNA takes on a different orientation relative to the sugar backbone than what you see in bDNA. And so this causes zDNA to have this kind of, I don't know, like very unesthetically appealing, wiggling, zigzagging geometry. You know, it's not a particularly prominent form of DNA, but it is thought to exist transiently in very short stretches of up to 100 base pairs, sometimes in the genomes, kind of like you see here. Now, an example, and if we were to zoom in on some zDNA here, and you were to look really closely, you would see, for example, that in that structure here, we have a syn conformation of this nucleotide here. So it's not completely an irrelevant conformation, but it is pretty rare and pretty minor. Now, the other thing about syn nucleotides to ask yourself is, well, a traditional Watson-Crick-Franklin base pair occurs when both nucleotides are in the anti-configuration. So if we have this interaction here between adenine and thymine, we've got our Watson-Crick-Franklin base pairs here between those groups, and the configurations are anti-anti. Is it possible for a syn nucleotide to form base pairs? Well, the answer is yes, assuming we allow for some funky geometries here. And indeed, you can see that if we place a thymine in this configuration here, you're able to again create a base pairing interaction between the amine here and the carbonyl, as well as a base pairing interaction between the lone pair of this nitrogen and the NH in the thymine. And these base pairs are a real thing. They are called Hoogsteen base pairs. And AT, CG, and even actually GG Hoogsteen base pairs are possible. And so it's not, these syn-anti interactions are, feasible, and they are sometimes observed. So what is different about these two base pairs beyond the fact that it's anti-anti versus syn-anti? Well, if we were to kind of take a closer look at which hydrogen bond acceptors and donors are involved, in a traditional Watson-Crick-Franklin base pairing interaction, we're seeing interactions between nitrogen 1 and nitrogen 3 of the adenine and thymine, whereas in this case, nitrogen 7, which is usually far away from the scene of a base pairing interaction, because we're in the syn conformation, in this lower Hoogsteen base pair, we're seeing an interaction between nitrogen 7 now and nitrogen 3 in the thymine. And of course, this is because of the different rotation about the glycosidic bond between the anti and syn conformations. Now, so where do you see Hoogsteen base pairs? So they're really very uncommon compared to Watson-Crick-Franklin base pairing. They do crop up in some structures because they allow for some very exotic DNA structures to form. For example, you can get 3 or 4 stranded nucleic acids, such as these things called G quadruplexes. So these are certain nucleic acid structures that you find in things like telomeres and stuff like that. And if you were to look closely in this structure, you would see an example of a GG Hoogsteen base pairing interaction right here. And these structures also sometimes form, or Hoogsteen base pairs sometimes form rather, when DNA is damaged or when DNA is bound by drugs. And so they have both kind of damage responsive roles, but it is thought that in some cases these structures may be important for gene regulation. Now, double-stranded nucleic acid structures are not the only type of nucleic acid structures that form. And in fact, many times RNAs, a single-stranded RNA, will end up also forming lots of base pairing interactions, secondary structure, and even tertiary structure. And an easy way to see that is that single-stranded nucleic acids could fold up on themselves to form secondary structure. So in kind of the simplest example we can think of, we have a single-stranded DNA or RNA molecule here. And if the sequence is such that if the structure were to fold back on itself and be able to form base pairing interactions, then you would get within a single-stranded structure regions in which there is base pairing interactions that would form helical secondary structure. And one important thing to realize is that this has the same sort of 5' to 3' base pairing interactions that we're talking about. That's why the structure needs to sort of double back on itself, because we're going 5' to 3' in this direction and 5' to 3' in this direction to form these base pairing interactions. And these type of interactions can occur both for DNA molecules as well as RNA molecules. Now, within the cell, we most commonly see these types of single-stranded secondary structures occurring in RNAs, and there's lots of examples of really important functional RNAs inside the cell that contain lots and lots of this secondary structure. And so a very famous one, for example, is called transfer RNA. These are involved in the process of translation in which protein synthesis occurs. And these RNA structures form from a single stranded molecule. They form a number of base pairing interactions to sort of adopt this kind of clover leaf like base pairing interactions that you see here. And what's interesting about many of these RNAs is that secondary structure enables the formation of more complex tertiary structure in a manner kind of similar to what you learned about protein secondary structure facilitating assembly of protein tertiary structure. And so these regions of secondary structure, so the base pairing in here, the base pairing in here, the base pairing in here, can then be kind of folded up and connected together to create a more complex three-dimensional structure. And while this is possible with DNA molecules, in general, in the cell, we're mostly going to see this as something that RNAs do. And I just want to mention that these structures can also become extremely complicated, and there's a lot of people in UW-Madison that study the kind of amazing things that RNA structures can do. So some of these RNAs can perform chemical reactions, and they have beautiful three-dimensional structures like you see here. And I just wanted to mention that so that you can kind of appreciate that although we tend to, and for most of the course, we'll be talking about RNAs in the context of simply kind of using the base pairing interactions to drive what's going on, it is important to remember that these molecules can form very complex structures that are kind of important for their function. Okay, so to sum up what we've talked about all throughout lecture one, I'm just going to go through this and make sure, double down on what I think you need to know. So we talked about how DNA is the genetic molecule of the cell. That's where the genes are, and that's the structure that's copied and passed down. Nucleic acids in general are made up of nucleotides that contain a base, a ribose sugar, and a phosphate, and those nucleotides are connected together in a sugar phosphodiester backbone. Nucleic acids can be single-stranded or multi-stranded, and in base pairing interactions, base stacking can stabilize polynucleotide structures. So in the double helix, for example, that base stacking is really what drives the stability of the helix. It is interactions between purines and pyrimidines that form base pairing interactions via hydrogen bonds. And different conformations of the nucleotide can affect the types of hydrogen bonding that you see versus, for example, in those syn antinucleotides that we saw, or the sugar pucker of those nucleotides can affect the overall kind of three-dimensional structure that we saw. So, for example, endoconformations that promote B-form DNA versus A-form RNA-DNA duplexes. We talked about how DNA is a typically double-stranded molecule inside cells, and the two strands form an anti-parallel right-handed helix. And we talked about how RNA is typically a single-stranded molecule, but by folding back on itself, it can form a wide range of secondary structures built from base pairing interactions, and those secondary structures can assemble further into more complicated three-dimensional structures as well. And so things that I think you really should be able to do from this lecture is you really need to know the chemical structure of each nucleotide base. And like I said, there's a companion video that I kind of walk you through how I would internalize that information if that's helpful for you. You need to know the types of bonds that a nucleotide is built up of. So, for example, phosphodiester bonds connecting the different monomers together in the nucleic acid polymer, and glycosidic bonds which connect the nucleobases to the sugar. You should also be able to identify on those nucleobases the hydrogen bond donors and acceptors, and which ones are involved in base pairing. It's really critical that you know that there is a polarity to the DNA polymer, and that DNA base pairing interactions occur when the strands are anti-parallel. So these single-stranded nucleotides will have some 5' to 3' end, and when base pairing interactions occur, the two strands are anti-parallel to one another. You need to know the structure of the nucleotides, the sugar pucker, that can determine the distances between phosphates to give you A form and B form DNA, and you need to know what a syn and an anti-nucleotide look like. And then finally, you need to know how nucleotides are added to a growing chain, and that is if the polymer is running 5' to 3', the 3' hydroxyl will be the site of new nucleotide addition. And so we say that synthesis occurs in the 5' to 3' direction. And it's also important for you to know the chemical mechanism by which that occurs. That is, you're going to have base catalyzed activation of a hydroxyl, which will then perform nucleophilic attack on your nucleotide monomer. And so I hope all of that's clear. And so just feel free to reach out if you need clarification on any of these issues. And next time we're going to be learning more about the structure of DNA and how we package it into chromosomes.